All right, thank you so much for the introduction, and thank you to the EMSL organizers for inviting me. It's really been an amazing conference so far, so much to learn about all kinds of different techniques. Today I'm going to tell you a little bit about what my group does, combining ion mobility mass spectrometry experiments with molecular dynamics simulations, as well as collision theory, to try to learn more about the structure of protein complexes. So as you might have kind of gathered yesterday or from your knowledge of mass spectrometry already, various techniques in mass spectrometry can reveal a lot about all the way from primary up to quaternary structure, composition, and stoichiometry of native biomolecular complexes. And I would say it really clearly, without a question, excels at primary structure, at learning information about exact mass kind of information, composition, stoichiometry. In between those, learning about secondary, tertiary, quaternary structure can be a lot more challenging, although a lot of the information you get from mass spectrometry pairs very well with X-ray, cryo-EM, NMR, and MD simulation data to help bridge that gap. What we're kind of wondering in my group is, can we do a little better in the native mass spectrometry world and try to learn a bit more about the structural information we can get from collision cross-sections, as well as use gas phase unfolding and dissociation techniques to probe quaternary and maybe even tertiary structure of protein complexes. So one of the fundamental questions that underlies native ion mobility mass spectrometry, where we take intact biomolecular complexes from buffered aqueous solution and we use electrospray ionization to put them in the gas phase of a mass spectrometer, is what happens to the structure of those ions as we remove all of that solvent and co-solutes. So to answer this question, we used MD simulations to try to understand what happens to a library of 17 different proteins and protein complexes that I've listed here that are used as standards in native ion mobility mass spec. And these span a very large range of sizes from about 2.8 daltons for melatonin up to 336 kilodaltons for the glutamate dehydrogenase hexamer that I've pictured here. So how do you get at the large scale structure of biomolecular complexes in the gas phase? Well, as you may have heard a bit yesterday, ion mobility is a great tool. We can measure the collision cross-section of ions in the gas phase, which is sort of like the average silhouette of the ion. So that's kind of a one-dimensional piece of information. It doesn't give us a lot of information about the detailed structure. And typically, if you want to use a cross-section to learn about structure, you need to compare it to condensed phase or simulated structures. For all of the work that I'll be showing you here today, we use the trajectory method for calculating collision cross-sections. This is a physically explicit model that tries to model the scattering of gas molecules as they impinge on the ions in our experiments. And we do that using a cross-section calculator our group developed called Kaleidoscope. So when we apply Kaleidoscope to that library of 17 IMMS standards and compare them to the experimental results that have been measured, what we find is large deviations between the structure or between the size of the ions that's computed versus the size that's measured experimentally, up to about 22% for some of those proteins, whether it's measured in nitrogen or helium buffer gas. So you can clearly see something's going on with the ions as they're transferred to the gas phase. To try to get at what exactly is going on with these ions, what we did was an approach where we looked at 5 different commonly used molecular dynamics force fields. These are force fields that anyone can access and that have often been used in ion mobility mass spectrometry, but have rarely been compared one against another. So our strategy to understand what is common among their predictions of compaction was to start with relaxation of the crystal structures gotten from the protein data bank with any missing residues added, relaxing those in water, placing charges in a favorable configuration on their surface, energy minimizing, and using a relatively simple in vacuo MD relaxation method at 298 Kelvin. And then after we've finished that computation, we do the collision cross-section computation in either nitrogen or helium. So let's take a look at what's common between what all of the force fields do. So all 5 of these force fields do predict significant compaction of ions in the gas phase. What I've got shown here is the PDB structure of bovine serum albumin And here's the result we get after compacting it with 5 of these different force fields. And we see they all compacted up to a pretty large extent, with the Gromos force fields compacting the most, and Opals and Charm compacting the least. So next we tried to take apart a bit more of this information. What exactly is being compacted in these ions? So there's been, for quite a long time, speculation, and going back to Breuker and McLafferty, that when you put biomolecular ions in the gas phase, essentially dangly amino acids that would be well solvated in solution are desperate to find polar groups on the surface of the protein to solvate them, so they essentially collapse down to the surface. So that motivated us to define the surface of our ions as any residues with greater than or equal to 30% solvent accessibility. That's using the Swiss Prot definition of surface residues. And we found out that all 5 of these force fields do show that the surface compacts exactly by this mechanism, where polar and charged groups are self-solvating on the surface, and in doing so kind of collapse down. So the Gromos and amber force fields predict the greatest degree of that, with Opals and Charm predicting it to a lesser extent. What about the rest of the protein? So we defined the rest of the protein as the quote-unquote interior, and we used the computed collision cross-section of that interior as kind of a stand-in for what's going on with its structure and compaction. So as I'll be showing you in the next couple slides, the green structure I've shown here in mesh is the original crystal structure, and the purple solid structure here, that's after the gas phase compaction simulation. So what we found out was that Gromos and amber force fields again predict a greater degree of compaction of the interior, essentially slight rearrangements of the various amino acids as compared to Opals and Charm. And by comparison, here's an example. This is Gromos 43A2 over here, and a much smaller degree of compaction of the interior is shown by Opals over here on the right. So we wanted to take a look in a little bit more detail at secondary and tertiary structure. Here's an example with beta-lactoglobulin, which is really nice because it has both beta sheets and some alpha helices. And what we found out was that all 5 of the force fields predict a slight decrease in both beta sheet and alpha helical structure, where alpha helical structure seems to be a bit easier to lose upon desiccation. You can kind of see the difference between some of the alpha helices here that have started to unwind a little bit over here on the right in our in vacuo MD simulations, but beta sheets seem to be a bit more robust. And this is really encouraging because it's long been a question of how well transfer to the gas phase preserves secondary structure. And this is consonant with, for example, spectroscopic studies done by Bowers and von Helden. We also looked at large scale changes, such as closure of cavities and grooves in proteins and protein complexes. We found out that all 5 force fields preserve large types of cavities and grooves. For example, in serum amyloid P component pentamer here, the large greater than 25 angstrom channel essentially in the center is kept through all of the simulations, whereas all of the force fields tend to close off holes that are smaller than about 5 angstroms. For mid-range holes, as we're calling them, between about 5 and 25 angstroms, the Gromos and amber force fields show a much greater degree of closure, whereas Charm and Opals show a lesser degree. So you might be wondering which of these is actually performing best as compared to experiment. Well, fortunately, with the 17 proteins we looked at, there was a clear winner, Gromos 9643A2 in nitrogen, especially, which is the buffer gas that most people are using nowadays to measure ion mobility for large protein complexes. We typically got 0% plus or minus 4% standard deviation away from the experimental values when we use this protocol. Opals and Charm, I would point out, are considerably worse, and another Gromos force field also performed relatively well. So we applied this method as well to look at results from the literature for charge-reduced proteins as well as membrane proteins. And we found out we got essentially the same ordering or the same type of performance, again with the Gromos 43A2 force field getting within 0 plus or minus 4% of the experimental values. That was good enough for us to apply it to the study of Staphylococcus aureus alpha-Hemolysin Heptamer and Hexamer pores, a bacterial pore-forming toxin that we study in my lab, where we were delighted to find that the experimental results were very much consonant with modeled structures that we did using this protocol for the pore forms of the toxins that we generated via electrospray ionization from detergent micelles. So we next asked the question, what happens if we look at the entire range of native charge states that are generated for protein ions in the gas phase? So as you probably saw yesterday in the native mass spec tutorial, typically in native mass spec, you generate an envelope of charge states, and it's long been a question of whether the structures at the high end of those charge states are really identical to the structures at the low end. And we can study that via collision cross-section a bit. One thing to consider, however, is that the collision cross-section that you should see for an ion, if two ions have identical structures but different charge states, the one with the higher charge state should cause more long-range scattering, which will tend to increase the collision cross-section. But if, on the other hand, one of them is more compacted, compaction should decrease the short-range component of the collision cross-section. So what happens when you kind of combine these two things together? Well, experimentally, it's pretty clear as day that across a wide range of protein sizes and charge states, we see that a number of proteins have very flat trends in their collision cross-section with respect to charge. So apparently these two effects are canceling out. So we wanted to see if we could recapitulate that with our MD simulations, modeling them with different charge states through the relaxation process. And indeed, we do see that it predicts very flat trends with respect to charge state, very similar to experiment. And what I should point out is that, especially for the smaller proteins, the range of cross sections explored by these native ions in our MD simulations is much smaller than the range you would predict if you assumed that they all had identical structures and only differed by charge state. So now that we have a handle on compaction of ions as we transfer them to the gas phase, what might we be able to learn in the future about tertiary and quaternary structure from unfolding and dissociation? So I'll just point out that protein unfolding has a long history in solution for learning about protein structure. It's been long known that the rates of unfolding of native-like proteins in solution in denaturation experiments, those rates of unfolding do correlate with the number of chain links or amino acids that are being unfolded. So there is information contained in the delta G, delta H, and delta S double dagger of unfolding that can tell us about the size or type of region that's unfolding. So we'd like to be able to do that in the gas phase, especially for proteins and protein complexes where we may not have the luxury of having a crystal structure already, for example. So the Rutlow group has really pioneered this technique of collision-induced unfolding, where you take a native ion in the gas phase, you subject it to collisions with a neutral buffer gas like argon, and you watch it unfold with ion mobility. And one of the things they've already noted is that the number of transitions that they observe for many monomeric proteins essentially matches the number of folding domains in those proteins known from solution. However, the mechanism of that unfolding, what drives that unfolding, is not known, and it's a bit difficult, therefore, to access information about tertiary and quaternary structure. So to better understand what is going on here and what we are trying to solve in my group, we should talk about what the unknowns in a typical collision-induced unfolding analysis are. So right here, what I've got plotted is the collision cross-section of bovine serum albumin, native ion, as we increase the energy of the collisions that it's undergoing in the gas phase inside the instrument, which cause it to unfold to these well-defined states that you see here. So in particular, the collision cross-section axis, that's again just that one-dimensional number. We typically don't know what the unfolded structures are that we are reaching, but as we've seen, properly chosen force fields with molecular dynamics simulations may help us a lot in that direction. Likewise, typically what is reported as the energy axis is the lab frame collision energy. That's just the charge of the ion times the acceleration potential that's being used to unfold it. So we don't actually know what the ion's effective temperature is to try to understand rate constants and barrier thermodynamics. We also typically are not analyzing in a quantitative way the shape of transitions between two different states as one decays and the other grows in. So we'd like to study both of those in my group to try to give us a more quantitative understanding of structure from CIU. So we modeled this in my group using a Monte Carlo collision model of our own development, where we model the collision and heating process as the ion is injected into the collision region at tens to thousands of electron volts of lab frame kinetic energy, undergoes hundreds to thousands of collisions with argon gas, which causes its internal temperature to go up, and it starts to unfold or dissociate, and then the reaction is quickly quenched by the helium cell of the instrument immediately before we measure the ion mobility cross-section. So as a summary of what we see there, the ion kinetic energy quickly drops as it starts to undergo collisions, and what happens is its temperature does go up and very slowly decays. And now we have, from our modeling, a profile of the temperature as a function of time, which we can put into things like the Eyring equation to get out the barrier thermodynamics for this unfolding. So I should point out that our simulations, first of all, indicated that remarkably over a huge range of sizes for the ions we're studying, the efficiency of energy deposition in collision-induced dissociation is nearly flat with respect to mass. It's about 26% efficient. So that tells you what temperatures you're achieving inside your ions during the collision process. So now we can use that information to study the shapes of those CIU transitions. So here's a zoom in of a typical collision-induced unfolding transition, where again, we're looking at bovine serum albumin in its native state, and we're adding energy to unfold it to a more unfolded state. And we can essentially take the ratio of the remaining unfolded, or the remaining native ion, and we can linearize that into a form that we can plug into either an Arrhenius rate equation or an Eyring rate equation to get at barrier information. We validated this method against a tried and true gold star method for measuring gas phase thermodynamics, which is blackbody infrared radiative dissociation, for 3 different proteins to study their dissociation. So myoglobin loss of heme and dissociation of shigatoxin and streptavidin. And what we found out is that our ion mobility-mass spectrometry based method matches spot on with these gold standard literature results. Now, one of the advantages I'll point out here is that we're doing this on a time of flight instrument. So all we have to do is adjust the collision voltage. We don't have to heat up the entire instrument like we do in a bird experiment. So our experiments take tens of minutes, not days, to acquire this kind of information. And we also, because these proteins are large enough, don't have to do a detailed vibrational mode or RRKM analysis. So this offers an exciting opportunity to use ion mobility-mass spectrometry because we don't have to use dissociation, i.e. a change of mass, as our readout anymore. We can actually just look at a change of collision cross-section, like you'd see in a collision-induced unfolding plot. So that's exactly what we did for a library of protein standards, as you see indicated down here. And we were able to use our Eyring model to derive the barrier enthalpies and entropies in both positive and negative ion mode. One of the interesting things we found out is that ions are indeed harder to unfold in negative ion mode. That agrees very well with results from Bush and coworkers. We also see an interesting correlation between the delta H and delta S double dagger of unfolding. And we were able to plot the Gibbs free energy of unfolding as a function of charge state, where we see very clearly sort of a 1 over charge type behavior. All of these results are telling us that Coulomb repulsion plays a very large role in the unfolding barrier height. And interestingly, these ranges of delta G double dagger compare very well to results from Page and coworkers for moving a proton from a highly basic side chain where it might start out in the ESI process to a less favorable amide oxygen to kick off the unfolding process. So we hypothesize that what we are seeing here is a common mechanism for unfolding of these ions in the gas phase via proton hopping. So putting this all together, this is kind of my conclusion slide. We now have much tighter constraints based on accurate compaction modeling and CCS computation for the y-axes of these plots. We can replace the lab frame collision energies with internal temperature on the x-axis. And by looking at the shape of the transitions we see in a CIU plot, we can get at the barrier thermodynamics. So what we hope is putting those things all together with modeling, like we did earlier with MD simulations, we can get much better informed models of what might be happening to these ions in the gas phase and how we can learn about their tertiary and quaternary structure. So with that, I'd like to thank you for your attention. I'd be happy to take a question.